this is the review for our final exam. So what I'm going to do is I'm just going to have our study guides that we went over all posted up on the screen. I'll have a slide where they're just blank and you can try to answer those out loud, and then I'll go on and answer those questions for us in the following slide. Now, if you can answer all these questions out loud, you'll be totally fine. And if you just want to listen to the answers, that's okay too. We're just going to talk through these one at a time. So going way back in time, first we had our scientific notation. So you can pause here and we'll talk about this in the next slide. So remember that writing in scientific notation is a way to make huge numbers, like numbers with a whole bunch, 20, 30 zeros, to compress them down into small little chunks. And it goes the other way too. We can make numbers that are really, really small that have a decimal point and then 20 or 30 zeros. We can smash those and make those way more compressed, more digestible, easier to look at. It's just really a way to be more efficient and concise in our writing. A number in scientific notation has three main parts. It's got a base, which is usually 10. It's got a coefficient, which is usually the first number in a number in scientific notation. And then that third piece is an exponent. And that can be really anything we want. Now remember, I gave you guys a quick and easy rule for remembering how to convert from scientific notation. Now, if you remember the rule and you know it's for converting from scientific notation, all you've got to remember is to flip the rule if you want to convert to scientific notation. So our rules over here apply when a number, it's already smashed into something like I have at the bottom of the screen here, the 1 times 10 to the 6th power. It's already smashed there and you want to spread it out. That's where this rule applies. If the exponent is negative, move the decimal to the left. And remember, there's an E in negative and an E in left. I've made those into huge letters on the screen. And if the exponent is positive, you move the decimal to the right. I in positive, I in right. So if you can kind of use that to help yourself remember, that'll be awesome. And so then there at the bottom, I have some examples of numbers in scientific notation and then out of scientific notation. Just be able to convert those. Just be able to move your decimal point. That's really what converting to and from scientific notation is about. Being able to choose the right way to move the decimal point. There may be a question or two on unit conversion, but it will be small enough and easy enough that you won't need a calculator for it. You'll be able to do it in your head or just move a decimal, and you don't have to worry about memorizing those conversion factors. So you don't have to know that 1 inch is 2.5 centimeters. You don't have to know that a centimeter is 0.4 inches. I'll give you those conversion factors. And the same thing if it's a pound or a kilogram conversion. I'll give you those numbers. All you have to decide is, do you multiply or do you divide to get the right number? In the second week, we went over scientific method. Here are our questions there. You can pause, try to answer them. We'll go on and answer them in the next slide. So I gave you 6 parts of a scientific method, and other sources will give you more points or less points. Just know these 6 from me and be able to recognize the correct order, be able to put them in order, know what each part is. So in class, we talked about using the scientific method on a bear or something. So the first part of the scientific method, you have observation. You observe something in nature. You observe a bear roaming about in the forest. The second thing is you have a question about it. You say, what is the bear doing? What do bears eat? What do bears do? Anything like that. You have your question. Once you have your question, you research. You find out, have other people already experimented and got the answer to my question? Because you don't want to go out and do a whole experiment if you don't have to. So you research, you collect data, figure out what we already know about the topic. After that, that's when you form your hypothesis. And remember, our hypothesis is a prediction of what will happen when we go to experiment. So you hypothesize that I think if I poke a bear with a stick, it will bite my friend in the arm, or if I poke a bear with a stick, it will run away. You can have any number of hypotheses. And that 5th point is, okay, now you actually have to carry out the experiment. You made your hypothesis, you predicted, now you got to experiment. You poke the bear with a stick, or you run your chemistry project. That's the experimental part. Then, once you find out what happened in your experiment, that's when you report and conclude. You write down, I poked the bear with the stick and it turned out it actually bit me in the arm instead of my friend. I didn't go according to plan. Or you write a report of how did your chemistry experiment turn out. But you always want to finish your experiment, write down what you found, and share it, even if it's just sharing it with your friends. Remember that the main difference in a question and a hypothesis is that the hypothesis includes a prediction. It has a predictive component. You can also think of it as it has an if-then component. If I poke a bear, then it will bite my friend. But a question is just a one part. It's what is the bear doing? What is happening out there in the world? It's just one part, no prediction. Hypothesis has the prediction. Also recall that the scientific method can't be used if something is unobservable or untestable, both directly and indirectly. And we talked a little bit about proving something indirectly using a scientific method. I want to go over it again because I felt like I didn't do as well of a job as I would like in describing this. So, we use the example of the black hole. Now remember, black holes are invisible. And around now, people are actually starting to take some pictures of them with fancy space telescopes, but assume for the moment that they're still invisible. You cannot see the black hole. It's invisible. You can't put it in a test tube. You can't touch it. You can't see it. What are you going to do if you want to prove that it exists? You have to look at the way it's affecting things around it. So when scientists started to get the idea of the black hole, it's not because they could see it. It's because there were random planets around it. There we've got a nice random planet on the screen, and it's being pulled towards something by gravity, and it's orbiting something. But we can't see what it is. It looks like there's nothing there. Why is this planet randomly orbiting? That's where they got the idea that maybe there is something there. We just can't see it. It's so dense and heavy that we can't look at it or touch it or get close enough to it. But we can look at the way it's affecting the planets around it. That's how the idea of the black hole came around. So they didn't test the black hole directly. They tested it indirectly by looking at what it was affecting around it. It was affecting a gravitational pull on the planets around it. And that's an example of using indirect science to prove something. So next we have our organic molecules. I'll let you look over these questions real quick. So a molecule that's organic has mostly carbon. Now this isn't the same as organic stuff in the store, even though that's probably going to contain a lot of carbon too, but they are different. And remember the root word in organic. It's the same root word in organism, because most living things are carbon-based organisms. Our four main macromolecules, we've got proteins, lipids or fats, carbohydrates, and nucleic acids. And those are made up of different monomers and polymers. So a monomer, remember, is one part. Mono is one, mer is part. And when enough monomers get together, they make a polymer. Poly means many, mer means part again, so it's many parts. So let's look at our table for the different monomers and polymers of each of the macromolecules. So for protein, we have the monomer as amino acids. When enough of those amino acids get together, they make a polypeptide, fancy word for a protein. For carbohydrates, the monomer is monosaccharide. I like that one because it's got mono in the name. You can kind of remember it. Then when monosaccharides combine and get together, they make the polymers, which are polysaccharides and disaccharides. Lipids. Monomers are fatty acid and glycerol. And when those come together, they make polymers of diglycerides and triglycerides. And last is nucleic acids. The monomers, the single parts, are nucleotides. When all those get together, we have DNA and RNA. So we had 1 week in lab where there wasn't a report or a study guide, and it was Plant and Animal Cell Day, Microscope Day. So what I asked you guys to do was to be able to recognize and give functions of the main structures in a plant and animal cell. And I'm going to go over those real quick with you just so you know what I expect. So just starting around 12 o'clock there, we have ribosomes. Remember that ribosomes are in charge of making proteins. They synthesize proteins. And you can see those ribosomes are coming down and they're attaching on this structure called the endoplasmic reticulum. What you want to know there is that an endoplasmic reticulum is going to function based on whether it's got ribosomes or not. If it has ribosomes on it, it's going to be a rough endoplasmic reticulum, and it's still going to work with making proteins. Now, all those pieces of the endoplasmic reticulum on the left there, you can see there's nothing on them. It's smooth. Smooth endoplasmic reticulum makes lipids. I always remember that as there's a nice smooth endoplasmic reticulum and makes a nice, smooth, shiny, slippery, nasty lipid. Those go together. Okay, so going back up, around 1 o'clock, we have the membrane. And remember, on an animal cell, it's a little more jelly-like. It's a plasma membrane. It's made up of fats, and it's going to selectively let things in and out of the cells. Just to the right of that, we have cytoplasm, and that gives the cell a little bit of structure, but it also makes sure that all those different structures, organelles in the cell aren't just rattling around in an empty cell. And there are some other processes that go on in there, glycolysis, but for the most part, no, it gives structure. Glycolysis happens in there, and it allows the organelles to be suspended in kind of a liquid. Down further, we have peroxisome. We didn't really talk about that, so I don't expect you to know the function. The nucleus, of course, stores DNA for the cell. The mitochondria is going to make energy for the cell. It's going to split down different foods into ATP. Right above that, we have the centriole, and the centriole is just involved with cell division. That's as complicated as I want you to get in your understanding of that structure. The top left corner, there's the Golgi apparatus, and it's in charge of packaging molecules, packaging proteins and other stuff, and getting ready to move them around the cell or send them out of the cell. So you can already see there are little circles around kind of the pancake stack structure. That's those packed molecules getting ready to be moved around. And just to the right of that, we have lysosome. The lysosome is in charge of digesting and breaking down things inside the cell. And so if you remember that word lys, L-Y-S, always remember that that means to split. There are a lot of words with that root word in there. Lys, lytic, or lysis, those are all the same. So there's lipolysis and glycolysis. Lipolysis is splitting fats. Glycolysis is splitting sugars. Anything with lys in it means to split or to break. The plant cell, just remember that there are a few extra bonus structures in the plant cell. We have the chloroplast, which is in charge of photosynthesis. Remember that animal cells break down food to get energy out, whereas plant cells will make their own energy using sunlight, and that's going to be done through that chloroplast structure. Plant cells also, generally, in the generic cell, they've got more of a centralized vacuole. We didn't talk about vacuole in the animal cell on the previous slide, but animal cells will have vacuoles, but the plant cells are going to have more of a generalized central plant cell or vacuole, and that's where they're going to store stuff. Plant cells like to store water, but they can really store food or water in vacuoles. Also, our plant cell has a cell wall, a little more rigid than the animal cell's plasma membrane, because plant cells, they're a little more stationary. They can afford some more rigidity. And so if you remember, that's what we punched through when we did our DNA extraction. So be able to recognize the structures and functions of the plant cell as well, and also be able to tell those main differences between the animal cell and the plant cell. For microscope, just remember the basic rule of microscope is to get total magnification. You want to multiply the eyepiece, those things you look in that your eyes touch, by the objective lens. And the objective lens is what points down towards the microscope slide that you're looking at. Here's our study guide for metabolism. Let you guys try to answer those. So when we talked about metabolism, we know that the main gland responsible for regulating metabolism is the thyroid. Incredibly important in the body. And other glands act on the thyroid, kind of give it some info, tell it what to do. Those two that I want you to know are hypothalamus and pituitary. They tell the thyroid, hey, release a certain hormone, or things along those lines. basal metabolic rate of an individual, this is the number of calories you need to stay alive and maintain your body weight if you were to lie on the bed staring at the ceiling doing nothing. Now, of course, that's not most people, but we move around, we have work, you have activity, exercise, things like that that burn extra calories, but you have a base level that these are the calories you absolutely need to stay alive to maintain your body weight if you're doing nothing else. And that's why it's called a basal metabolic rate. It's the base number. Now, there are three factors. Well, really, there are a ton, but the ones I want you to know, the factors that can increase that basal metabolic rate are, the first one is age. The older you are, metabolism tends to slow down as you go along. The second factor is genetics. Some people just hit the genetic lottery, can eat whatever they want and just burn through it, not gain any weight. Other people so much as look at a salad and they gain 5 pounds. So genetics is that second piece. And then the last piece is just body mass. The more cells you have, the more calories you're going to need to maintain all of those cells, to maintain that body weight. Then we moved on to respiration. Here are our questions. Aerobic respiration is the process of converting food into energy with oxygen. So that's with oxygen. And then anaerobic, same general idea. We're still converting food into energy, but we're not using oxygen, and it's way less efficient. When yeast performs anaerobic respiration, it makes ethanol. That's called fermentation. As far as our aerobic respiration formula goes, be able to recognize the correct formula. Remember that we start with the sugar, and with oxygen, we split that sugar, and we get carbon dioxide, water, and really important for us as organisms is the energy piece. Don't forget about the energy piece. That's the whole point of breaking down the sugar. We want to get the energy out of that molecule. Then we went on to photosynthesis. Now, photosynthesis is the process that plants go through to convert sunlight, water, and carbon dioxide into food. So they're doing the opposite of what animal cells are doing. Animal cells are breaking the food down. Plant cells are making food in the process of photosynthesis. Also be able to recognize the formula for photosynthesis. And if you look at it, you'll see that if you just take the aerobic respiration formula and reverse it and change the energy from ATP to sunlight, you've already got your photosynthesis formula. It's just aerobic respiration in reverse. That structure that performs photosynthesis, that's going to be the chloroplast, which we just mentioned a second ago with our plant cells. Okay, a little bigger study guide here. This was the combined weeks on DNA replication and genetics. Here are our questions. I'll let you look through those. So the central dogma of biology, or called central dogma of microbiology, that's the rule that DNA turns into RNA and RNA turns into protein. You may hear people say that DNA will code for RNA, RNA will code for protein. That's fine and dandy too. Just know that DNA goes into RNA, which turns into protein. Remember that when DNA is turned into RNA, it's called transcription. And then when RNA goes on to and is turned into protein, it's called translation. And I always remember which one comes first by just putting them in alphabetical order. There's a C in transcription, and that comes before the L in translation. So I know that transcription comes first. So remember that for genotype and phenotype, the genotype is the underlying code, that genetics of the organism that you can't see with your naked eye. And the phenotype, that's the physical expression of the genes, of the genotype. And you can see that with the unaided eye. That's the overall picture. I always remember that phenotype, physical expression. They both have F sounds in the beginning of the word. And so you can put those together and try to remember that. The main structure in the cell that synthesizes proteins is ribosomes. We mentioned that just a minute ago. Moving on, make sure you can tell me the 3 RNA subgroups, even though you don't have to really tell me their function. We've got tRNA, which is transfer RNA, rRNA, or ribosomal RNA, and then mRNA, or messenger RNA. When it comes to the dominance and the recessiveness of genetic traits, the dominant trait is the one that will be expressed in the phenotype of the organism. So even if you've got a trait for something, if it isn't dominant, it may not be expressed. An easy way to think about this is like blue eyes and brown eyes. Now, I might have one trait for each one. I might have a blue eye trait and a brown eye trait. But the blue eye trait is recessive and the brown eye trait is dominant. So even though I have one of each, the brown eye is going to dominate over the blue eye, and then the brown eye is going to be expressed in the phenotype. So mutations. So a mutation is any error that a cell makes when it's making more DNA or dividing or doing some other reaction. Most of those things, those mutations, not good. 1% or less of all mutations are beneficial to the organism. 1% or less help the organism to be better fit for its environment. The rest, either neutral, and the most of them are going to be harmful. A lot of mutations just end up killing cells, especially if it's just a single cell type of organism. Enzymes. So an enzyme is a molecule, it's usually a protein, not always, but usually, and it reduces the activation energy for reactions in the cell. And I always think of this as a coupon. You get the same cellular reaction, but 70% off the energy. And that's really efficient. Cells can't do a lot of the things that they would be able to do if they don't have enzymes. They're critical for any kind of cell reaction. Moving on to blood typing. I'll let you guys answer these. So remember our rhesus factor in blood, it's a protein in blood, and it determines if your overall blood type is positive or negative. So you've got, your main blood types. You have your A, your B, AB, and O. But then the rhesus factor determines, well, are you AB negative? Are you AB positive? Are you O negative, O positive, so on and so forth. That's really important for blood transfusions and things along those lines. O negative is going to be our universal donor. Loves to give things to people. And then AB positive is going to be our universal recipient. Loves to take. Antigens and antibodies. So an antigen is a foreign substance. It can be bacteria, something like that, that comes into the body and activates the immune system. It triggers an immune response. So when an antigen comes in and triggers that response, our body makes antibodies. These are the good guys. An antibody is a protein, usually Y-shaped, that the immune system makes in response to an antigen. So antigen comes in, we make antibodies, antibodies go and beat up the antigen. Really good guys there. Okay, we're on to natural selection. Here are our questions. So natural selection is the process by which the organisms best fit for the environment survive. And when they survive, they get to pass on their genes to the next generation. Charles Darwin is credited with the idea for natural selection. And another name for that is survival of the fittest. Also remember that natural selection is not the same. It's different from macroevolution, because natural selection kind of happens all the time. Some organisms are better fit for the environment, and so they get to pass on their genes and they determine the genetic structure of a little ecosystem. But macroevolution, it requires change in domains or kinds of organisms. So I have to go from non-life to bacteria to reptile to bird to mammal, so on and so forth. And there's really just no scientific or historical evidence of anything like that ever happening. Now, there are three big laws of science that the theory of macroevolution violates. Really, it violates more, but these are the three that I want you to know. The first one is the law of biogenesis. I know you guys talked about Louis Pasteur and him disproving this idea of spontaneous generation with his experiments. He showed that if you're going to have a living organism, living bacteria, anything that's alive has to come from something that was already alive. Life must come from life. Life cannot come from non-life. That's our law of biogenesis. However, somewhere along the line, the theory of macroevolution requires that there was no life, and then poof, somehow life just generates out of the nothingness. It violates that law. Our second law is Newton's third law, or the law of cause and effect. And the Big Bang is an effect without a cause. Even if you can explain how a period-sized ball of matter came to exist, there's no logical, rational, scientific explanation for why it would randomly explode. Things don't randomly explode in the universe without some other underlying cause. So that's why it violates the third law. And then finally, we have the first law of thermodynamics, or the law of conservation of energy. If you guys are in chemistry, you know this, that matter and energy, they cannot be created, they can't be destroyed. You can change their forms a little bit. You can change them from matter to energy, one to the other, but you can't create them out of the nothingness. Somewhere along the lines, the theory of macroevolution requires that there was once nothing, and then out of the nothingness, matter appeared. Matter and or energy appeared. That just violates this law. It doesn't make scientific sense. Those are the three laws I want you to know. And we are on to the last, DNA extraction. Here are our questions here. Remember that DNA is going to be acidic. That's what the A in DNA stands for. To get DNA out of a plant cell, you want to punch through the cell wall. It's kind of more rigid. If you think back, we just talked about it. Plant cell, a little more rigid than the animal cell. You got to break through that tough exterior. And then the DNA, both plant and animal cells, is going to be stored in the nucleus. That is the end of this review. If you guys can answer all these questions, you're going to do just fine. Thanks for listening.